Today, we're honored to have Mary Relling give today's lecture. Dr. Relling earned her undergraduate degree in pharmacy from the University of Arizona and her doctorate in pharmacy from the University of Utah. She completed postdoctoral fellowship training with Bill Evans at St. Jude and Urs Meyer at the University of Basel. She joined St. Jude as a faculty member in 1988 and in 2003 was named chair of the Department of Pharmaceutical Science. She is also professor at the University of Tennessee, both the College of Medicine and Pharmacy. Her primary research interests are in the treatment and pharmacogenetics of childhood leukemia and clinical implementation of pharmacogenetic testing. Dr. Relling is chair of the NIH's Pharmacogenetic Research Network and co-founder of CPIC, the Clinical Pharmacogenetic Implementation Consortium. She was elected to the Institute of Medicine in 2009. Please enjoy the presentation.
Okay, welcome. My name is Mary Relling, and I am the chair of the pharmaceutical department at St. Jude Children's Research Hospital in Memphis. I also co-lead CPIC, the Clinical Pharmacogenetics Implementation Consortium, with Terry Klein of Stanford, and I'll be talking about this resource throughout my talk today, which is on clinical pharmacogenomics testing. Actionable pharmacogenetic gene-drug pairs have been known for a very long time. Since about 1950, it's been known that by altering the dose of primaquine in patients who are G6PD deficient, one could avoid serious adverse reactions, and several other gene-drug pairs have really been worked out as long ago as 3 decades ago. And many more continue to be discovered in the present day. So what do I mean by actionable? For example, the data that relate to TPMT and thiopurines are particularly strong. Back in the late 1980s, before thiopurine doses were adjusted based on thiopurine methyltransferase genotype, 100% of patients who are homozygous deficient for this gene product had life-threatening myelosuppression. About 35% of those heterozygous for low-function alleles had life-threatening myelosuppression compared to only about 7% of the majority of patients who are homozygous for normal function alleles for TPMT. And subsequently, in follow-up studies, we started adjusting the dose of thiopurines based on TPMT genotype, and by doing that, we had the same amount of myelosuppression, a much lower amount of higher myelosuppression in those individuals who were heterozygous for low-function alleles versus the majority of patients who were homozygous for normal function alleles. And the other very important piece of evidence that leads us to be confident using TPMT status for adjusting thiopurine doses is that the risk of relapse was not higher in the patients that were heterozygote for low-function alleles shown in red compared to the majority of the population that was homozygous for normal function alleles shown in black, even though those patients that were heterozygous for TPMT status received lower doses of thiopurines. So if we can reduce toxicity without compromising efficacy, then that's exactly the kind of actionability on pharmacogenetic testing that will be comfortable moving forward in the clinic. Another example of something that is frequently used in the clinic is screening for HLA-B variants prior to prescribing abacavir. And in this New England Journal paper from about 10 years ago, but it was shown that by prospectively screening HIV-positive patients before making the decision about prescribing abacavir, they could avoid clinically diagnosed hypersensitivity reactions or immunologically confirmed hypersensitivity reactions substantially by using a patient's HLA-B genotype to withhold abacavir in that minority of the population that has a high-risk HLA-B allele. So these are the kinds of gene-drug pairs that have risen to the level of clinical actionability, but for the most part, the use of these pharmacogenetic tests in the clinic is still rare. In fact, a recent survey of pharmacies in the hospital setting in the U.S. found that the vast majority of hospitals did not offer pharmacogenetic testing. Only 7% offered pharmacogenetic testing, and that was up slightly from 2.7% in 2009. So you can see that genomically informed prescribing, even in the hospital setting, which probably is more advanced than the outpatient setting, remains underutilized. Another piece of evidence showing the low uptake of the clinical use of pharmacogenomic tests is provided in this summary from a large academic medical center. These were medical centers associated with the University of Washington in Seattle. And it was, however, it was a setting in which there weren't any directed efforts at preemptive genotyping for pharmacogenetic tests. It involved over 130,000 patients and over 3 million hospital or clinic visits. And they looked at medication orders for 95 drugs that had germline pharmacogenetic testing mentioned in their FDA-approved drug labels, about half of which were considered actionable by CPIC. And they found that less than 1% of the prescriptions for those 95 drugs were accompanied by any pharmacogenetic testing. And when they limited their analysis not to just mention of pharmacogenetic testing in the FDA label, but FDA actually recommending or requiring pharmacogenetic tests, it only rose to 1.5%. So this gives some idea, in this paper just published a couple months ago, again, that pharmacogenetic tests are really underutilized. This has been reviewed recently by the Dutch Pharmacogenomics Consortium, and they reviewed 12 programs, primarily in the United States, but a couple in Europe as well, that are doing some kind of preemptive strategy for implementing pharmacogenomics. And this is an increase from a few years ago, but again, the vast majority of major pharmacogenomic testing implementation programs are centered in a few academic sites. Back in 2009, before we started CPIC, we did a survey of pharmacogenetic experts. These were members of the Pharmacogenomics Research Network and of the American Society for Clinical Pharmacology and Therapeutics. And we asked them what their challenges were to implementing pharmacogenetics, because these were people who had a stake in trying to implement pharmacogenetics in the clinic. And 95% of respondents said that the process required to translate genetic information into clinical actions was a major barrier. And the next two most frequent responses were genotype test interpretation, so how to move from the genotype to the phenotype, and also which gene drug pairs should be selected to implement first. In fact, this last barrier is one that I think has become even more challenging in the last few years. This is just a sampling of the many, many direct-to-consumer, direct-to-clinician, for-profit pharmacogenetic testing companies that are putting a lot of pressure on patients and on clinicians to order pharmacogenetic tests. And sometimes they're pressuring to order these pharmacogenetic tests for gene drug pairs for which the evidence is really not adequate to have clinical actionability. So not only is it important for clinicians to know which gene drug pairs to implement, but also to know which gene drug pairs not to implement. I'll talk about our work at St. Jude because we're approaching clinical implementation on two fronts. As I mentioned, we have co-formed CPIC with our colleagues at Stanford, and this is an international consortium of experts to facilitate clinical implementation. And really, we did this somewhat selfishly because at about the same time, we started a protocol here at St. Jude called PGen for Kids, the goal of which is to implement preemptive pharmacogenetic testing for patients. And so we needed to have these kinds of clinical guidelines, international guidelines, to do our own clinical implementation. CPIC's website is indicated here. And as I mentioned, it's an international consortium primarily composed of volunteers and a small group of dedicated staff. who are trying to facilitate use of pharmacogenetic tests for patient care. And we have members from all over the world, I think probably over 300 members at this point. And what we do is create very specific gene drug guidelines, clinical guidelines. And these guidelines are designed to help clinicians understand how to use available genetic test results to optimize drug therapy. It's a pretty important assumption of CPIC that it's not whether pharmacogenetic tests must be ordered, because we believe that we will be moving from the current status of ordering primarily gene-specific tests to someday it will be true that many people will have their entire genomes or exomes sequenced, and then the challenge for clinicians is not whether the test should be ordered, but how to use the genetic test results that have been generated to utilize them to improve prescribing. And so this has been a key assumption that helps CPIC avoid questions of things like cost effectiveness. Again, it's if the question is not whether to order the test, but how to use genomic information to inform prescribing, it's a completely different set of considerations. Our CPIC guidelines are posted on our website, and they capitalize on PharmGKB resources. They're freely available, no limits on use, peer-reviewed with CPT having the first right of refusal, and they have standardized formats and a minimum set of elements. We have standardized grading of evidence and of recommendations. We can update them as needed on the CPIC website without waiting for an updated publication. We have a conflict of interest policy, and we closely follow IOM best practices for clinical guideline development. And this is just a screenshot of a few of the front pages of the about 20 CPIC guidelines that have been published so far. So indicated here are the genes that are the subject of CPIC guidelines and the applicable drugs that are covered by those CPIC guidelines. In progress right now is the tamoxifen CYP2D6 guideline, which we hope will be submitted this month, as well as a CYP2B6 efavirenz and an inhaled anesthetics guideline. So again, the guidelines are available on the website, and every guideline has its own homepage, and at the top of each one of the guidelines homepages are any updates since the publication. And this has been a useful way of showing our users how to keep up with new information. So one of the most utilized web pages on CPIC is this gene drug page. And I think that's because it provides the community with a grading of gene drug pairs to get a quick assessment of whether those are deemed to be likely to be actionable and whether they will be the subject of CPIC guidelines. So we do have an algorithm by which we evaluate gene drug pairs. If the gene is already subject to a CPIC guideline, then again, one can imagine clinicians are really faced with a very strong imperative to decide, are there other drugs besides the one already subject to a CPIC guideline that are actionable? And we consider each drug gene pair to be in 4 levels. CPIC level A or B is that a prescribing action is recommended at some level of strength. Based on the gene drug pair, as well as, very importantly, the evidence that supports the alternative therapy to be used for individuals that have a high-risk genetic status and are prescribed a high-risk drug. And examples of these would be TPMT and thiopurines or warfarin and its associated genes. CPIC level C is one in which no prescribing changes are recommended based on genetics, and that could be because the alternatives are unclear or the evidence is weak. And this is especially true for genes for which testing is common for other CPIC level A or B genes. A good example of this is CYP2D6 and propranolol. There's no question that CYP2D6 metabolizes propranolol, but because of the involvement of other enzymes and because of the way that the drug is titrated, there's really no actionability that is recommended based on CYP2D6 status and propranolol. And then, of course, there are many, many publications that link a gene to a drug. PharmGKB does a good job of annotating these publications, and these are ones for which there's publications, but which the evidence is definitely weak, alternatives are unclear, and testing may be rare. So an example of this might be GSTM1 and cisplatin. So the way that gene-drug pairs get to be evaluated is by them being actionable in other professional society guidelines, nominated by a CPIC member, a high level of annotation on PharmGKB, and we're always continually evaluating gene-drug pairs and giving them a provisional status as CPIC level A, B, C, or D. We have a grading system for the prescribing recommendations, which is a simple system where there's strong, moderate, or optional prescribing recommendations. And all of these are considered to be potentially actionable, especially in the situation where the genotyping has been done preemptively. We also have a fourth category that we added in the last year or two where there is no recommendation. We really try to avoid writing guidelines where there's no recommendation for any genotype for any drug. However, there are examples where there's just insufficient evidence or confidence to provide a recommendation to guide clinical practice, and I'll give you an example of that in a moment. So here is an example where this is ondansetron and tropizatron, two antiemetics that are definitely metabolized by CYP2D6. And CYP2D6 can be classified into four main phenotypes: ultra-rapid, normal, intermediate, and poor metabolizer. And there is evidence that for those rare 2% of the population that are ultra-rapid metabolizers, that they metabolize those drugs so quickly that an alternative antiemetic agent should be considered, and that is a moderate recommendation. And for normal metabolizers, there's no evidence that prescribing need be informed by CYP2D6 status. That's a strong recommendation. For intermediate and poor metabolizers, even though it seemed to make intuitive sense that poor metabolizers might be at increased risk of some of the toxicities associated with these antiemetics, such as QRS prolongation, there's actually data to suggest data addressing this topic and not finding any evidence to support any dosage change for poor metabolizers. Although the rationale certainly seems plausible that such individuals might be at increased risk for toxicity. So the guideline authors voted to have no recommendation for intermediate or poor metabolizers. So this is an example of a guideline where there may be multiple type strengths of recommendation depending on the phenotype and the drugs. Overall, this gene-drug pair page gets many views. We've had 30,000 page views in the last year. It covers 352 gene-drug pairs, 46 of which are level A, 99 level B, 83 level C, and the biggest chunk are level D gene-drug pairs. So although this is always in flux, and it would require a full literature review to definitively decide on a level, this provides clinicians with one of the things that they identified as a barrier, and that is to help prioritize which gene-drug pairs to implement. We think that the number of gene-drug pairs that are going to be actionable in the foreseeable future is relatively small. So we've estimated the workload for the foreseeable future and estimate that there will be 31 genes covered by CPIC guidelines, and that would be involving about 40 CPIC guidelines overall that need to be written. So far, I think we have 20 that are published or in press. And of course, many of the genes that are actionable for some drugs are not actionable for other drugs. So is this going to revolutionize prescribing? Does pharmacogenomics affect all drugs? If we think that there's something like 1,200 chemical entities that are approved as drugs in the US, and there's something like 18,000 genes, we have to acknowledge that the number of gene-drug pairs where there's going to be actionability accounts for only a minority. About 7% of FDA-approved medications are covered by the about 90 drugs that we think will be covered by the CPIC guidelines. And this does make up a slightly larger percentage of all prescriptions in the U.S. because some of these drugs are extremely widely used, maybe as many as 18% of outpatient prescriptions in the U.S. But still, the likelihood that a gene defect is going to have essentially monogenic effects on a drug, making it actionable in the clinic, is small, and we neither want to oversell or undersell the importance of pharmacogenomic testing for prescribing in general. The way in which guidelines or gene-drug pairs are prioritized includes a number of different elements. Is there prescribing actionability? What's the severity of the clinical consequences if genetics aren't used to inform prescribing? Is there an available genetic test for that gene? How commonly used are the drugs, or how common is the genetic testing? How common are the high-risk genetic variants? Is there mention of genetic testing in the drug labeling? And are there pharmacogenetically based prescribing recommendations from other organizations? So all of these go into how we are constantly reprioritizing which gene-drug pairs to tackle and prioritize first. We have a Gantt chart that indicates all of the gene-drug pairs that have been identified as level A and B genes, and even a few level C genes that we think are very important for having guidelines available if possible. So these would include things like factor V Leiden and estrogen use, which is, again, very heavily marketed to clinicians and to patients, but which most experts agree the evidence is not clear enough to allow prescribing recommendations. So having negative guidelines for some of these gene-drug pairs might be useful. We've surveyed our members and have them reprioritize gene-drug pairs that aren't currently the subject of CPIC guidelines. And when we did that, we found that 2C19 and proton pump inhibitors came to the top of the list. And another gene-drug pair that is of a lot of interest, especially to the public, is CYP2D6 and ADHD drugs, which, again, there's heavy marketing to pediatricians and directly to consumers, claiming a benefit for that when in actuality there's probably very little actionability there. So as I mentioned, we try to update the guidelines when important new information comes out, at least to make users aware of that. So here's an example for ivacaftor and CFTR, where the FDA approved additional CFTR variants that should benefit from the use of ivacaftor from the original approval of the drug several years earlier. And so just adding variants is something that can be easily done online. One of the challenges in clinical implementation, and I guess in many different areas of trying to change medicine, is maintaining links with all of the players in the community. So there are many different NIH-supported groups that are working on implementation of genetic testing in the clinic. There's professional societies such as ACMG and ASHG that are involved, as well as pharmacologic groups at the FDA. There's groups involved in the medical record business that provides the infrastructure by which most of these pharmacogenetic tests are going to be rolled out. So there's quite a bit of effort involved in maintaining links and trying to avoid duplication of effort. Resources page on CPIC provides a list of individuals who are using CPIC guidelines to do clinical implementation of pharmacogenomic testing, and this provides a way for investigators and clinicians to find each other. And now I'll kind of move into how CPIC relates to a real implementation project, using our implementation project at St. Jude, PGen for Kids, as the example, where our long-term goal is to have preemptive pharmacogenetic testing adopted as the standard of care for all St. Jude patients, and our goal is to implement all CPIC guidelines. This is a protocol that we started in 2011. We basically try to enroll every patient at St. Jude who might receive any drugs, and the only exclusion criteria are patients who've received a prior allogeneic or liver transplant. Allogeneic because the blood from which we would extract their DNA and even the buccal cells will be primarily from donor, and liver transplant because so many actionable pharmacogenes are genes that are expressed primarily in liver. Our process is that we do obtain informed consent. We get a blood sample. We send the sample for genotyping, which covers around 230 genes, and the vast majority of those genes stay in a research database. And the whole idea of the protocol is to selectively put results into the EHR as we develop clinical decision support that informs our clinicians about how to use that information for prescribing for at least one drug. And then we follow up with patient education. These are the gene-drug pairs that we've implemented since we opened in 2011. We started with TPMT and CYP2D6, then SLC01B1, CYP2C19, DPYD, UGT1A1, 3A5, NUT-T15, and our most recent is CYP2C9. And this just looks at the same data in a different way, showing that 22 drugs have been implemented as linked to these 9 genes, and sometimes we're just adding new drugs for a gene that's already been implemented at St. Jude. If we look at these 9 genes and 22 drugs, by the first 4,000 or so patients that we've studied, the percentage of individuals who will have a high-risk genotype for each one of those genes is indicated here. So for some of them, it's extremely low, like 0.4% for DPYD, and for other genes like 2C19, it's extremely common to have an actionable diplotype. In fact, if we look at all of the patients that we have, it's a very small percentage of patients who have no high-risk genotypes, and there are a fair percentage of patients who have as many as 5 high-risk genotypes. Overall, 90% of patients have at least one high-risk genotype in those first 3,800 patients, and we can extrapolate based on the known frequencies of the actionable pharmacogenes that this will increase to 99% once we implement all the first 12 or 13 actionable CPIC genes. We've enrolled more than 4,000 patients, or we've approached more than 4,000 patients for enrollment. 94% were enrolled on the protocol. A special consideration is in pediatrics is that when children reach 18 years of age, they have to be reconsented at the age of majority, and 99.5% of kids who turn 18 want to stay on the study. And out of all these patients, we've had 2 incidental findings where a pharmacogene predisposed to a disease risk, and both of those cases were Klinefelters. Because we're studying G6PD, which is an X-linked gene, we occasionally have boys that have an extra X chromosome, and that's obvious from looking at their G6PD genotype. So how do we get from genotype to interruptive clinical decision support for prescribing? How do we go from these raw genotype data with the position of the genome and the variant that's present to a very text-based, understandable, point-of-care alerting prescribers how to change their prescribing? And I'll talk you through how we do that and how CPIC guidelines help us to do that. So if we look at the CPIC guideline for voriconazole and 2C19 as an example, here we're going from genotypes to alleles. And so we have tables that translate the raw genotypes into CYP2C19 alleles, and we assign function to those alleles. And these are done by these gene-specific tables that are linked to each of the CPIC genes maintained on PharmGKB. Every row is an allele here for CYP2C19. Every column is a nucleotide position, which is mapped to the human genome using 5 different systems of mapping. So there's unequivocal identification of which alleles are which. And then we use standardized terms to assign function to each of the alleles for that gene. And these are updated online as needed. So then we need to go from alleles to diplotypes because everybody has generally 2 copies of each gene. And this is particularly important in pharmacogenes as compared to other areas of genetics. In other areas of disease-related genetics, the question is often just who has the variant or not, whereas for pharmacogenes, it's usually very important to distinguish between individuals homozygous for variant alleles versus heterozygous for variant alleles. And listed here with the yeses are those actionable genes for which it's important, for which the prescribing recommendations are completely different based on diplotype compared to just the presence or absence of the genes. So we have to go from the genotype or sequencing data to haplotypes and diplotypes. Now at St. Jude, we've been using the DMET array for several years, and actually the software associated with these arrays is very helpful. Every row is an individual variant position, but it does come up with a called diplotype based on the likely frequencies and phasing of the variants that are present in the genome. And we've checked, and that phasing works very well in the AFI software. To specifically address this challenge that's going to be becoming more and more common, that sequencing of whole genomes is going to be done, PharmGKB has partnered with Marilyn Ritchie and others to develop software called PharmCat, which will go from VCF files, the variant files that are generated from sequencing, to generate the haplotypes or diplotypes that are important for the CPIC guidelines and generate a report that corresponds to the CPIC guideline prescribing recommendations. And there are many other tools that are being developed to try to help users go from genotype to phenotype. So we have to go from diplotypes to phenotypes, and we have to be able to interpret those phenotypes in light of drug therapy. So again, those gene-specific information tables posted along with every CPIC guideline. Here's the diplotypes for CYP2C19. You can imagine that for genes like 2C19 or especially 2D6, these tables are thousands and thousands and thousands of rows long with the possible phenotype associated with each of those diplotypes indicated: intermediate, normal, rapid, poor metabolizer, as well as some kind of indicator whether that's considered an abnormal, actionable result versus a normal or routine result. How we translate that at St. Jude in our PGen for Kids study is that we have created a pharmacogenetics tab in our EHR. That's important because it's not encounter specific. Most genetic tests are lifelong tests, and so you don't want to have to dig through dates of different DNA samples. You want to be able to look for all the DNA samples for a patient. What are the pharmacogenetic test results that have been generated? And there's a consult that's associated with every gene test result, and that provides a passive level of CDS for clinicians who want to look at pharmacogenetic test results and see what the passive static interpretation is for that. So here's an example of for a CYP2C19 rapid metabolizer diplotype and how that is interpreted. Also, now we have some consults that are based on the results of more than one gene. So for example, for thiopurines, we need that consult to include the genetic test results from both NED-T15 and TPMT. So we bring both gene tests together under one consult that is reachable by linking to either one of those gene test results by themselves. We've created a system for building templates for these consults based on the sections that we think should be included in every pharmacogenetic test result. That is the phenotype assignment, the diplotype interpretation, the phenotype interpretation, and a quick review of medications that might be affected, some basic prescribing recommendations, and an educational link for people who want to learn more. And we put together those consults based on software that we've built called Consult Builder. So here we are pulling down the gene of interest, CYP2C19. We can pull down the diplotype of interest from a pull-down menu. And then for every one of those set of diplotypes, we have some text sentences that apply to the interpretation of the diplotype that will go into each applicable consult. And this is just showing that consult deconstructed into those color-coded forms. This has also been useful because we want the exact same information to populate the St. Jude formulary, which is our go-to resource for our clinicians for medications. And so if a clinician at St. Jude goes to the formulary, they will see that there is information on pharmacogenetics for all the applicable drugs. There's also a place where they can just see all of the therapeutic guidelines, including pharmacogenetic guidelines. And by clicking on that hyperlink, they can see all of the pharmacogenetic guidelines that apply to medications that we use at St. Jude, and all of the associated information that might be available for them. Again, the same language that's used to populate the interruptive alerts, the consults, is also used to populate the formulary. Again, someone can look up, here they're looking up CYP2C9 and celecoxib, and they can see what the dosing recommendations would be for a poor metabolizer for 2C9 if the clinician wants to consider prescribing celecoxib. So this actionability is the bottom line that we get to of how to act on pharmacogenetic test results. And again, every CPIC guideline now has example language that can be used to interpret the different phenotypes for each of the actionable genes and download it into an EHR system. We also provide algorithms for how to go through each result for each gene that's the subject for a pharmacogenetic CPIC guideline. And I'll talk for a second about our interruptive alerts, which is a form of active clinical decision support. We divide them into two main categories. The first is the pre-test alert. So if a patient is prescribed a high-risk pharmacogenetic drug, how does one decide whether a genetic test has already been ordered on that patient? And if there is no pharmacogenetic test result on that patient, how does one alert the prescriber? And the second is the post-test situation where the test result has been generated. So there we just need to be sure that if the test result is high risk and a high-risk drug is prescribed for that patient, that an interruptive alert fires at the clinician. So these pretest alerts, this is an example of one for CYP2D6 and codeine, which is, again, very important in a pediatric hospital where we really should not use codeine unless we know the CYP2D6 status of the patient. And it not only alerts the prescriber that there is no 2D6 genotype test yet on this patient, but it allows them to click a box and order the test right then if they want to. Now, the other is this post-test result alert, which fires if there is a high-risk test result already present for the patient. And here we say, based on this genotype, the result, the patient is predicted to be a poor metabolizer. We give them alternatives that they can use to codeine and how they can consult with a clinical pharmacist to get more information if desired. The way that we handle firing alerts based on high-risk pharmacogenetic test results at St. Jude is to create a problem list entry based on the high-risk genotype. That problem list entry resides with things like the underlying disease. So here, the presence of hepatocellular carcinoma, and this patient has 2 high-risk pharmacogenetic test results. It's an automated process to go from the test result to the problem list entry, and then we create CDS that drives off of the problem list entry. Of course, it could be done directly from the high-risk test result. One disadvantage of doing that is if the test result is generated by an external lab, more and more patients are coming in with their own genetic testing results from direct-to-consumer testing companies. We still need to have a way of driving CDS off of high-risk test results no matter where they come from. So that's why we've elected to do it from the problem list instead of directly from the genetic test result. So another principle is that we're trying to incorporate non-genetic information into some of these interruptive alerts. For example, for voriconazole, not only is 2C19 important, but so is the route of administration, oral versus parenteral, and so is age. Kids less than 12 need a different dose than kids greater than 12, regardless of their CYP2C19 status. So here where we're firing the alert, not only are we taking into account age and route and genotype, but we're also telling the prescriber we're taking it into account. And we found that this is also a big challenge in implementing genetic tests. Prescribers need to know what was considered when we fire these alerts at them. They don't like seeing alerts that they don't understand what the basis for the alert is. And this is another example of how these alerts are having to get a little more complicated for thiopurines. We're having to incorporate both the TPMT and the NUD-T15 genotype result into the alert to tell the prescriber that that's how we've come up with the recommended doses for that patient. Another challenge that we've run into, and we knew that this would be likely, is that we have implemented genes at St. Jude. Even when we know there are other drugs affected, we may not have built the CDS for every single affected drug at the time the gene goes into the EHR. And that can result in us missing important interactions. So an example of this happened within the last few months here. We had an 11-year-old boy with leukemia who was late in therapy. He was having intermittent thrombocytopenia, episodes of hematemesis, and was started on omeprazole. After he had more hematemesis, endoscopy showed that he had developed esophageal varices. And the outside physician who was treating this patient asked us for a consultation because the liver enzymes were increased. But we looked in the St. Jude EHR, found that the patient was an ultra-rapid metabolizer of CYP2C19. That diplotype was in the EHR, but we hadn't built yet any CDS for proton pump inhibitors. And so the clinician had no easy way of understanding that they might need to use a much greater dose than normal of omeprazole because we hadn't alerted them to this problem. Now, luckily, CPIC hasn't gotten around to writing a guideline for PPIs and CYP2C19, and St. Jude hasn't either, but the Dutch working group in pharmacogenetics has. And this information is easily available both in the publication and on PharmGKB, and indicates that 1 to 200% increase in dose is recommended for ultra-rapid metabolizers. And so in this case, that patient had a big increase in their PPI dose and eventually was doing fine. So the CDS that's needed for clinical actionability of genetic test results, I hope I've given you a flavor that there has to be a way to unambiguously and uniformly refer to genetic test results in order to build clinical decision support that can be useful for prescribing information. And for the most part, genetic test names and results and phenotypes are not standardized across healthcare systems. And therefore, EHR vendors are stating that it will be very difficult for them to build clinical decision support tools based on genetic tests if the community of clinicians and researchers doing pharmacogenetics can't do simple things like come to terms on standardized terms. So CPIC collaborated with many other groups in coming up with some standardized terms for pharmacogenetics, and these include allele function for all of the pharmacogenes, where we use increased, normal, decreased, no function, unknown function, or uncertain function as standardized ways of referring to allele function, and then standardized terms for phenotypes that differ if it's a drug metabolizing enzyme, a transporter, or a different type of high-risk gene like HLA-B. The other thing that having standardized terms will help with, in addition to driving CDS, is to try to detect accidental duplicate genetic testing. So it's been shown by the Mayo Clinic and others that unintentional duplicative genetic testing is becoming more and more common. And of course, not only is that a waste of resources, but it also results in the possibility that there will be disagreement between 2 different or 3 different tests for the same gene. So again, one would never be able to fire an alert to ask, do you really mean to order that second test of UGT1A1 or TPMT, or do you want to use the pre-existing result? No one would be able to make that determination unless we have standardized terms for referring to genetic test results. We also are highly engaged in patient education about pharmacogenetics, and we offer to send a letter to the patient's house for every gene test result as it migrates into their EHR. 98% of patients participating in PGen for Kids ask for having that information. Again, we have standardized templates for putting together these letters. And here's, for example, a CYP2D6 gene result has been put in your EHR. The majority of the population is here in green. You're here in red. You're a poor metabolizer. We give them a little bit of information about what that might mean and give them or their clinician a link and a phone number for more information if they would like it. So, of course, this isn't optimal, but given that we have a fragmented healthcare system and we don't have an underlying EHR that will allow these lifetime genetic tests to be used as a patient moves from system to system to system, this is one way that patients can inform all of their prescribers about their status. And this letter is posted in the EHR. And at St. Jude, we do have a patient portal, as most healthcare systems do now, so patients can directly access their genetic test results via this mechanism as well. Now, many others throughout the world are trying to come up with more electronically based, more systems that will require more action on the part of patients. So again, in the European Ubiquitous Pharmacogenomics group, they're coming up with a QR code card that can be scanned and then will directly access all of the updated pharmacogenetic information for that patient via any computer that can access their websites. So this may be something that can be done in the future, and we're looking into such systems at St. Jude as well. We have these patient information sheets that are publicly available to anyone. Some of them are drug specific, so like here's a patient medication sheet on codeine, and it talks about the fact that genetics can be important and refers them to another do you know sheet on CYP2D6, which they are also given every time they have a high-risk result or are prescribed a high-risk drug by one of our clinic staff. We're using pharmacogenetic testing as a metric for patient safety and quality. So TPMT-guided thiopurine dosing is one of those metrics. This is incorporated into our quality and patient safety team's institutional safety metrics, where our threshold goal is to have 100% of leukemia patients have their TPMT genotype known prior to initiating therapy at St. Jude. And you can see this has varied a little bit over the years, but for the last few quarters, we have been hitting our goal of 100% of patients having their TPMT status known before they get their first dose of thiopurine drug. CYP2D6 is another gene for which we would like to see the percentage of patients who have CYP2D6 results in their EHR go up over time, and this green line indicates that it has, and therefore the number of alerts that have had to fire, the pretest alerts that have had to fire, has gone down over time as a higher and higher percentage of our population are already genotyped before they get their first dose of codeine. And in fact, we showed that in our sickle cell population, there were no codeine prescriptions given to patients who had a high-risk, ultra-rapid, or poor metabolizer phenotype after a couple of years of implementing preemptive pharmacogenetic testing for all of our patients, including sickle cell. This process of rolling out each gene-drug pair, so every column here is a gene-drug pair at St. Jude, and every row is something that has to be done before we say hit the button and make it go live. And you can see these include things like evaluating the evidence and developing the CDS and getting all of our software and lookup tables and consults and patient letters and do you knows updated, and working with the patient education committee, working with our P&T committee. We have an oversight committee for the institution that reports to P&T, and they have to approve every gene drug pair before we roll it out and make it go live. So there's many, many steps that have to take place before we roll out a new gene drug pair, and this is why this process has taken some time. We share all of the information that we have on our website at PGen for Kids. So this includes a lot of information about the protocol. We update the website every time we roll out a new gene and every time we add a new drug to one of those genes. So there's a little bit of information that's available to both patients and clinicians. We have an educational video that shows patients and families talking about using pharmacogenetic tests, and that's available for anyone. All of our publications related to the study. We have competencies that our pharmacists complete as we roll out every gene drug pair. So depending on the level of practice of the pharmacist, they have some basic training in how to answer questions about pharmacogenetics and to make them aware of what pharmacogenetic testing is available. And we have a ASHP accredited PGY2 residency in pharmacogenetics, and these people are getting great training and going out into the world and helping to do implementation at other sites. And finally, links to some other resources. So I will stop there. I would like to acknowledge for CPIC, especially my co-PI, Terry Klein, and our collaborators at Stanford who are involved with CPIC. Our director of CPIC is Kelly Caudle, and she works here at St. Jude. And we have some other individuals here at St. Jude who also work extensively on developing CPIC guidelines and policies, as well as external members of CPIC who are volunteers and provide a tremendous amount of input. Our guideline authors play a critical role because without their expertise, our guidelines wouldn't have the peer-reviewed status and acceptance that they've gained in the clinical community. I'd also like to acknowledge all of my collaborators on the PGen for Kids protocol here locally at St. Jude that involve many members of our department, but also information sciences and representatives from all of the clinical departments and divisions at St. Jude, and my colleagues at Medical College of Wisconsin who have done our genotyping for us on this protocol. So I thank you for your attention. If you have any questions, please contact the program coordinator, and thank you.